Hey guys, my name is P. Nemes, and today I will be talking with you about genetics. Now, genetics is one of my favorite fields of classes that I've taken at my school because it does have to do with my major. My major is cellular and molecular biology at John Carroll, and genetics is kind of cool just because it's something that like many people don't know much about besides what they've heard about, in the media from like hearing reports about GM food or cloning or just certain things like that. But the three main areas that I wish to cover today with you all will be some of the mechanisms involved with genetics and some famous scientists, as well as some of the applications of genetics. Now, for the mechanisms, the central dogma of molecular biology, like pretty much encompasses like all that, which has three components. First of which is DNA replication. DNA replication is very important because this is the step where cells are going to make identical copies of their DNA and make sure that DNA is packaged into like the new cells that are dividing them. And this is the first process in the central dogma. The next transcription is the term used to describe the process where one DNA strand is used as a template to make one strand of RNA, single strand of RNA, which is called messenger RNA or shorthanded as mRNA. But this mRNA serves as like a precursor to protein synthesis, or in some cases actually the RNA, if it's not used to be coded into a protein, which I'll talk about next, will serve as like a functional RNA of some sort. But following transcription, you have translation, which is the last component in the central dogma. And this is about actually like synthesizing the protein from the messenger RNA. And proteins really serve as like the framework for like all the cell's functions, from like biochemical reactions to like structural support to like transforming like molecules across different like cell components and all that stuff. But all that is like all done by proteins, so it's really important. But two like very famous scientists who both ended up winning Nobel Prizes were Francis Crick and Carey Mullis. Like first, Francis Crick, he was the person who discovered the three-dimensional structure of the double helix like structure of DNA, and like several decades later, he also proposed the wobble position hypothesis, which that actually has to do with translation, because when the messenger RNA is being like read by this something called ribosome, the ribosome reads the sequence base pairs in like threes. So like Francis Crick proposed that the last one was like different than the first two. But Carey Mullis, who made his discoveries several decades later, he invented a technique that's still widely used today called PCR, polymerase chain reaction. And this tool is very important for anybody who's studying genes or the sequence of genes, because you can take like a few short to like medium length like sequences of DNA, run them through the PCR, and like within a couple hours, like you would have like millions and millions of copies of those like same sequences, which makes it very important, for just science people who study this. And Kerry Mullis claims to have made these discoveries based upon his uses of psychedelics in the 60s and 70s. And next, I want to talk to you guys about some of the applications of genetics and GM foods, genetically modified foods. That's something that most people, when they hear, like, they'll immediately think it's something bad or it's just something like a natural. But basically what it is the process of putting in genes that are not like endogenous, meaning they don't like naturally exist in that like specific, like, whatever it would be, a plant, animal, or whatever. So like when it naturally exists in that plant or animal's own genome, but some new gene has been like introduced into the genome, which expresses some kind of protein that serves, some kind of unique function for that cell. But doing this could really help combat world hunger and malnutrition. And contrary to what the majority of people probably think, like, it's not dangerous and it's been like tested extensively, so it's not, just something that's like just thrown out there. I mean, this stuff is like highly regulated, so. But it's just mainly the media that just like tries to put out that image of it. But GM food can do like a lot of good things for us, so I don't think it's something that should be discredited. And stem cells are just getting like more and more important, like really like in the field of medicine, especially. But these types of cells start out as in like non-specific cell, because like cells, not all cells in your body like are the same, but they do all have like the same like copy of DNA, the same exact sequences. But the reason why you have different like cell types, hence different like types of body tissues, is because like certain genes are like turned on and like turned off. So like certain genes are expressed, whereas certain genes are like not expressed throughout someone's own like growth and development. But these type of cells serve as huge like sources for treatments for people with cancers, like transplants, like organ transplants that might went low and they need to like have it redone or have a alternative like treatment. Certain diseases that like may cause someone to like lose function in one of their organs, like their liver or something, and they need a new one. Stem cells like serve as huge like potentials for that. And just to cap it up now, I'm glad I can inform you about genetics, and I'd like to thank you for watching.